You guys are doing a fantastic job. I do need to pause for a second though, and present our first polling question to our audience. Which I told you, did I not say at the top of the hour this is going to be the fastest 60 minutes that you spend all week? This question is for our audience. We're just curious what modalities are included in your biosimilar development plans. Antibodies, ADCs, cell therapies, oligos, peptides, vaccines, gene therapies, other. Let us know, and we'll revisit that poll response here in a second. I don't know if that poll went live accidentally already, Miles. If it did, okay. Well, if you haven't responded, do you get another chance?
Yeah, we'll push it out again.
We'll push it out again. Take another look. While we're waiting for that, and even though we sort of had a sneak peek, do our panelists have any predictions around which of these modalities might be considered the lowest hanging fruit for our audience? Kaushal, I'll start with you. What do you anticipate might be the top response here? I don't know if Kaushal's muted. I can't see Kaushal. There we go.
Yeah. So I think antibodies, I believe, is much better choice at this point.
Yeah.
So ADC is difficult actually. So many companies are developing, but I think MAPs we all have good experience with MAPs. So.
It looks like, let's go ahead and take a look. I think Kaushal's right. It looks like Celltrion paved the way, right? Antibodies are definitely the top selection here.
Right.
I want to talk a little bit about controlling costs. You know, Kaushal, you and I had a conversation a week or so ago about sort of the cost of litigation, the cost of, IP protection, and that being a very important factor in developers' plans or aspirations even to get into the biosimilar space and address some of these challenges that Julie and Hadel have been talking about. What are some of your thoughts around controlling the cost of getting into, getting a seat at the table in the biosimilars game?
So I believe that one of the key factor, so as Juliana said, it's almost 10 years plan. So it's definitely a very expensive. But in my view, you can definitely control the cost at various parameters, and with the advance of lot of new technologies nowadays, I'm seeing and I'm working with many of our partners who are supplying some sort of technology. So from the clone level, people are much serious about having their expression level. So as you are going to produce this molecule. So being a generic, the biggest challenge is that the cost and market penetration. So from the day one, if your clones are producing a much better expression and giving a high expression, probably you can definitely have a market benefit. Then coming to the other side of the technologies, I see people now do not follow a very routine technologies available, or our conventional technologies available on fermentation or upstream side, where there are new technologies where people try to use continuous manufacturing or other sort of technology. By that they can improve the not only the product quality, which is very important for regulatory perspective, but also the yield of the product. So finally, it really determines if there are 4 to 5 companies are competing for one products, and anyone who really use a recent technologies, latest technologies, would be definitely having a advantage over the other generic who are not using the latest technologies. Then coming to the patent litigation side, as I said, council should really work very close to the scientist and try to design rounds as well as resolve the IP issues in the beginning. That would definitely control litigation cost and time. Then important is in biosimilar, brand has no limitation. They can assert n number of patents. So we have seen in Humira, brand has asserted 60 patents, 74 patents. So litigating 60 and 74 patent is really, really very expensive. And I don't think so. Like most of companies can do that. Even we have seen like Boehringer and Amgen and every Sandoz, like all of them like settled with FB. So people has tried like invested a lot of time and money in litigation, but finally they just settle the way FB wanted them to settle. So the what is the way out? I believe is that working and designing around the patents from the beginning, and then focusing on the new technologies, as like Miss Ethel was telling about clinical side and like analytical side. So also we should focus on the development of new assays because now like like I believe like agencies are much open for. So there are a provision like type 1 type 2 meetings, and where you can every biosimilar applicant can propose and try to convince FDA with their new approach. By that they can reduce the time. So if they develop a new assay, they can convince FDA that I do not to conduct or perform this lengthy study, but I can show the similarity or the efficacy of the molecule by this
Mm.
assay. Or there are simulation methods, mathematical simulation methods are available that people are implementing in their means biosimilar development, which improve the confidence. And even before putting the molecule in the animal or human, they get better predictability, like the outcome, like in what way it should go out. So these all are like the tools. And then finally definitely litigation, like means hiring a right patent attorneys, experienced patent attorney having a relevant backgrounds are much always beneficial, because in US especially, like there are many law firms who claims biosimilar, but actually in reality I see like there are very few firms who really has a good experience of handling of biosimilars. And if you really work with them, probably like means you can have a better settlement and
Mm,
less time in litigation, and then finally you save the cost. So these are the
yeah.
factors I see is important. Yeah.
Kaushal, and while I've got you on the screen, Anna just asked how many reference batches should be tested and how many, which assays should be performed in order to prove biosimilarity. I think you were just kind of scratching the surface on that. Are there standards that you're aware of, or is it? I mean, is there a standard?
So see, I think like that's a great question, and I believe like I'm basically a patent attorney, but I have seen like almost like my in 18 years of experience, I have worked with many of all the type of scientists. And uh, it depends on what type of products you are picking up. Like so, suppose it's immunologic products, then I think Hetal and Juliana is more relevant for this question. But like if there are a products called like means having a rheumatoid arthritis, let's say, and there is a specific mechanism of the product is there, and you shows like how these products actually behave in the human body. So if you decide, if you develop some assays, you bring some cell lines, you develop the receptor in the cell line, the same receptor, and then you try to show the impact of the your biosimilar product, and then try to convince. So probably FDA may either reduce some of your studies or time period, or may waive off some of the portion. So it actually depends. So this question is actually basically depends on the types of product you select, and
Mm,
really the result what you actually get. So but people are really working hard on analytical side. They are bringing a lot of technologies in analytical to show the better characterization. Because if like the as far as I understood FDA guideline, they say like analytical burden is high. So if you prove the analytical, which is really a uphill task, I have not seen anyone who can wave off the human clinical so far, but the attempt is that to reduce the human clinical burden, so that you can speed up the development and provide the product at the better cost, because finally the clinical is taking a lot of time and the cost. So that's why people are attempting, and many of the companies even in US, I work with some companies, they provide simulation methods. You give them a molecule, they will develop a mathematical algorithm. They will tell you like this would work, whether your PK will match or not match, in what way your phase one should go. So this is my understanding on the assay and this kind of methods.